thanks to all for joining, and thanks to Karthik for organizing this seminar. So this is the second webinar in this series, and I'm really glad to be a part of it. And I'm sure such webinars will be conducted at the rate of once every month from various researchers around the globe who are working in the domain of pyrolysis technologies. So in today's presentation, so I'll be trying to make the presentation for around 40 minutes, 45 minutes. If you have any questions, as Karthik mentioned, please type it in the chat box, and towards the end, I'll be able to answer everything. And today's presentation is going to be on, it's going to be broadly on analytical pyrolysis for biomass conversion application. So before I go to my presentation. So this is yeah. So that's the broad interest of my research group. So it's simply summarized there. So we deal with a wide variety of feedstocks and processes and products. So typically we deal with thermochemical conversion of algae to biomass, coal, municipal solid waste that are very relevant in today's context for solid waste management. And we work on a wide variety of processes, as I said, thermochemical processes, including pyrolysis, gasification, combustion, and hydrothermal liquefaction. And we employ a wide variety of tools, including analytical pyrolysis, GCMS, analytical pyrolysis, FTIR, TGA, and a host of other catalysis techniques to probe the reactions, both for a fundamental understanding as well as for the applied application-oriented research. And we deal with a wide variety of products. Ultimately, we want to make sure that the products that we get out of all these processes and feedstocks are going to be really useful, and we finally make use of the energy or the biofuels or the chemicals in some form or the other. So with that, this is going to be the outline of my today's presentation, and this is going to be on. So I'll give a very broad introduction to fast pyrolysis, why it is important, and how pyrolysis GCMS or analytical pyrolysis evolved as a technique for characterization, and now it is being used for a wide variety of applications. I'll talk about the three different analytical pyrolysis systems that are available in the market, and luckily we also have them in our lab, so we have been trying to utilize them for multiple applications. And finally, I'll give one of our recent results wherein we try to do microalgae pyrolysis, using all these different micropyrolysers, and I'll be talking about what are the different real heating effects that really contribute to the observed product distribution. So while we are talking about fast pyrolysis, first, we all know that what is pyrolysis in itself. Pyrolysis is simple heating of the material in inert ambience to a high temperature of the order of 500, 400 to 600 degrees C. And that results in a wide variety of complicated products based on the nature of the feedstock. So in this figure, what the different transformations when you pyrolyze a biomass are shown here. So typically you take a biomass or a waste or a plastic, whatever be the material, right? That's in the solid phase, and then you apply a high temperature in inert ambience, and that results in initially the formation of what are called as primary liquids or aerosols. And then they can undergo a lot of other secondary reactions to form low molecular weight compounds like gases. It can form condensed oils, and the solid material forms what we call a secondary charge. So what is very important is pyrolysis is a process that involves all three phases: solid, liquid, and gaseous phase. And based on the pyrolysis severity, by pyrolysis severity, I really mean that you apply a very high temperature, or you allow the material for a long enough time at a high temperature, right? So it's an interplay between temperature and residence time, and that can actually result in change in the product distribution that you get. Okay, So as three different phases are involved, you get three different products. Okay, solid char, liquid oil or pyrolysis oil, and the gas phase, non-condensable gases, right? So based on the nature of the severity, right, the product distribution can vary, and hence you can actually decide what kind of products you want from the pyrolysis process. So this is a rough example, of biomass fast pyrolysis. So typically, I mean, this is like a formula for any common general biomass, right? So it results in typically, significant yield of the oil. You get char and gases, and of course, based on the composition, the product distribution will essentially vary. Okay, and in fast pyrolysis, by fast pyrolysis, we really mean that we apply heating at a very fast heating rate. So think of you have a sample and you are subjecting it to a high temperature in 1 second. Okay, but and in the other case you are actually heating it very slowly, right? So this really determines the kind of reactions and the interactions that occur at such conditions. Okay, so the bottom line is the residence time or the heating rate controls the product distribution. Okay, so we generally call something as fast pyrolysis, okay, when the heating rate beta is very high. Okay, so again, 1000 is just a number, but typically you want to heat the material instantaneously to a set temperature of, let's say, 400 to 700 degrees C, and you allow very less time in the reactor so that the products are simply carried away by the gas stream. So under this condition, you end up in significant amount of liquids. Okay, so this data is for pyrolysis of wood. Okay, that is reported in the literature. And now if you're going to, let's say you're not varying the temperature, but if you're varying the heating rate and also the residence time, right, you can see that the liquid yield is going to decrease, whereas the gaseous yield and char yields are going to increase, right? Similarly, as you further increase the residence time, reduce the heating rate, and even reduce the temperature, you can shift the product distribution from liquid to char. Okay, so if the material is derived from wood or any biomass, then it is called as a biochar, or it is called as simply char if you derive it from any plastic or any kind of solid waste. Okay, so by tuning the operating conditions, okay, temperature, residence time, or the heating rate, you can simply shift the product distribution, okay, from a liquid rich product distribution to a char or gas rich product distribution. And that is the versatility of pyrolysis, that is that it is being applied in a wide variety of instances. Okay, before I really go into analytical pyrolysis, okay, let me tell you what is the chronology of this analytical pyrolysis. Okay, so pyrolysis is a process in itself, but analytical pyrolysis is it is more like a technique that has evolved so that you can simply employ it to study a wide variety of things in the lab. Okay, so analytical pyrolysis, again, it probably started in 1948 or early early 1950s, right, when it was actually used for to study the pyrolysis of polymers. Okay, and remember, now we are talking about analytical pyrolysis for biomass conversion as a special case, but when it evolved, it was originally designed or developed for characterizing the polymers. Okay, let's say you synthesize a new polymer, and if you want to understand whether, if you want to understand what is the structure of the polymer, right? So you can actually use this analytical pyrolysis technique. It actually evolved as a polymer characterization tool and not to study the pyrolysis reactions. But of course, now we have actually adapted it mostly to understand the pyrolysis chemistries. Right. So initially people started with pyrolysis MS. Okay. So you have online pyrolysis MS and offline pyrolysis GC. That means you do the pyrolysis, take the products, inject into a GC and analyze what products are formed. Right. And you can see in 1964-66, you have Curie point pyrolysis reactors and pyrolysis capillary GCMS. That means you do pyrolysis. Okay. That is what is shown here. You do pyrolysis, take the pyrolysis products through a gas chromatograph. So you have a capillary column that will separate the products into different components, and you send it through the MS. So MS will tell you what component it is, what are the different components. It gives you the identity of the components, and you can even quantify the exact amount of the different components using MS. So it's simply an integration of pyrolyzer, GC, and MS. Okay, so that is analytical pyrolysis, and it's simply a lab scale setup so that you don't require significant amount of sample. Okay, and then you can simply screen a wide variety of materials for their compositions or characteristics and so on. And on the other side, you can see here you have you can see the development of the various the chromatography methods and mass spectrometry. So you can see that JAP, Journal of Analytical and Applied Pyrolysis, launched its first issue in 1979, and from then, right, you can actually see a wide variety of papers on analytical pyrolysis GCMS applied to a wide variety of materials, not just for synthetic polymers or biomass, but also to a lot of composites and functional materials. Okay, so when we talk about this analytical pyrolysis, what is very important is we are actually talking about small sample masses. Okay, we are not talking about taking a huge chunk of the material, subjecting it to pyrolysis and understanding the product distribution, but we are actually talking about very small particles, very small mass of the sample, which can be of the order of milligrams. Okay, so why we need to really go for small sample mass and small particle size is actually depicted in this figure. Okay, so this is a figure plot of bio number. Okay, so bio number is nothing but the time scale of conduction to convection, and the pyrolysis numbers, right? Okay, that's the time scale of reaction to conduction and reaction to convection. Whatever be the material that you want to pyrolyze, okay, you can actually find out these numbers based on their basic properties like specific heat, thermal conductivity, density, and so on, and actually determine what is this number and in which axis we are actually lying. Okay, so you can see a kinetically limited isothermal regime. Okay, wherein if you're going to take a material of particle size lesser than 20 microns, okay, or sometimes even 50 microns, right, you can actually lie in a kinetically controlled regime. Okay, that means reactions are going to be the slowest step, and that is going to really determine the typical product distribution. Whereas if you're going to take a very big particle, okay, let's say 1 centimeter or something like 10 centimeters, right, which are, of course, this is a regime that is very typically used in practical scenarios. Okay, under such conditions, the reaction is typically conduction or heat transfer limited. Okay, so that means there is going to be some gradients within the particle or within the sample, and that is going to result in different product distribution. So it is because of this fact that typical analytical reactors typically operate in a kinetically limited regime, and hence we use very low amount of sample, lesser than 1 milligram, and particle sizes which are very, very small, so that the particle is uniformly heated and the products essentially evolve out of the particle, and there are no heat transfer and mass transfer effects coming into the picture. Okay, coming to some of the applications, analytical pyrolysis GCMS is an excellent characterization tool. Okay, this is what I emphasized in the beginning. So here you can see the typical product distribution that you get when you take a polymethyl methacrylate polymer, neat polymethyl methacrylate polymer, and subject it to pyrolysis at 500 degrees C. Okay, so this is actually a well-known result, but I just want to emphasize this fact that you get this methyl methacrylate, which is the monomer of the polymer, in very high yields. Okay, so this is a typical GCMS total ion chromatogram. Okay, so this says that this is the methyl methacrylate peak, which comes at around 4.5 minutes. Right, then you have the product distribution from polystyrene and then ultra high molecular weight polyethylene. Okay, so this says that if you're going to pyrolyze the sample at a specific temperature, these are the products that are going to be evolved. And you can always do a calibration in your GC mass spec to find out the exact mass percentage of each of these different components coming from your sample. So typically, you can see all the monomeric peaks coming at the earlier retention time periods in your GC mass spec, and the dimers, trimers, tetramers, and long chain hydrocarbons, right, typically come at later time periods. Okay, so it's an excellent characterization tool, and probably this is a very simple application that I'm showing in this slide. Okay, coming to its evolution. Okay, I said that Py-GCMS evolved as a polymer characterization tool. Okay, you can actually see a lot of works from Suji and Otani. Okay, where they have actually pioneered this technique, and you can see here that how Py-GCMS can be used to understand the polymer microstructure. Okay, so this is the typical pyrograms in the tetramer region for polystyrene. So you have atactic polystyrene, isotactic polystyrene, syndiotactic polystyrene, and any natural commercial polystyrene sample. Okay, so you can see that the signals that you see in the total ion chromatogram, right? These do correspond to specific, racemo or a meso type of a sample. Okay, that is present in the tetramer. Okay, so this is called as a polymer microstructure. Generally, those who are aware of polymers, these are long chain components of repeating units. Okay, but how are these monomeric units repeating? Okay, and that really determines the microstructure of the sample. Okay, so you can see here that so this is the total ion chromatogram, and this is the mass spectrum of peak B. Okay, so if you click on this peak in a typical GCMS, you can see this typical MS of the peaks, and it really corresponds to the different fragments which correspond to the scission of various bonds present in your tetramer sample. Okay, so this example I would like to highlight that this is a very classic example of how Py-GCMS can be used to understand the structure of the polymer. Okay, so from this you can say that how many type of racemo, meso, dyads or triads are going to be present in the polymer sample that has been synthesized according to a particular technique. Another example, this is about the product distribution from head to tail and head to head linkages in a polystyrene sample. So typically, if you think of a polystyrene sample, you have head to tail linkages. So head is a bulky group and tail is a simple methylene group. Okay, so you can also have head to head linkages and tail to tail linkages. Okay, so when you do pyrolysis GCMS of any commercial sample which has head to head or head to tail linkages present in it, it actually gives you a different product distribution. Okay, so you can see that if you take polystyrene sample with head to tail linkages, okay, you see that you get 80% or 81% of styrene. Okay, whereas if you're going to pyrolyze a head to head polystyrene, that means a polystyrene sample having majority of head to head linkages in it, right? Then you see that your polystyrene is going to be significantly reduced, and you're going to get a wide variety of dimers, trimers, and tetramers that are coming out of your polystyrene sample. Okay, so this is what I would like to emphasize, that it is an excellent characterization tool. Okay, beyond much beyond its application in biomass research, I think it's an excellent characterization tool. Okay, if you have synthesized a new material or a new polymer, right, you can actually do a pyrolysis GCMS, right, and based on the product distribution and also correlating with other characterization techniques like NMR, right, you can pinpoint the exact microstructure or the exact sequence distribution of the various linkages present in your polymer sample. Okay, and I would like to say that those of you who are working on pyrolysis GCMS, this is a very good book. This is actually a database of Py-GCMS thermograms and MS of a wide variety of polymer samples, synthetic polymers to natural polymers and so on. And this can be used as a good reference if you're going to start working with polymer pyrolysis. So it gives you a good idea of what are the typical products you can expect, so that you can really correlate it with the actual products that you're getting. Khurshid, I think this book is not available freely. Of course, this is authored by Frontier Lab people, so maybe you can contact them and they can give you a copy or something. But this is a very good book. Okay, I'll show some examples before moving. So we have actually use Py-GC technique to understand the gamma aging of to understand the aging of polymeric samples. This example is about gamma aging of silicone rubber insulation, which is used in transformer insulations. Okay, so here you can see a typical thermogravimetric analysis of silicone rubber, virgin as well as aged silicone rubber, gamma aged silicone rubber. You can see that as you apply gamma radiation, right, you can see that the thermal stability of the polymer is actually going to decrease. Okay, this is what you can understand from a simple thermogravimetric analysis, which gives you the mass loss profile of the sample. But if you're now going to do a Py-GC of it, right, you can actually see what is the product distribution you get out of it. Okay, so this is the virgin sample. Okay, you can see this is a typical, I'm showing here, m is equal to 1, 2, 3 correspond to this. The amount, the cyclicity of the sample, whether it is a smaller cycle or a bigger cycle. So you can see that as the material is aged, right, you will see that the number of units in the cyclic ring of this silicone material is going to increase, right? And that really points to the fact that there is a significant degradation or scission and recombination of a lot of these different OSI bonds in the material when you're going to subject it to gamma radiation. Okay, and this is a typical product spectrum. I would probably suggest you to look at the red and the green boxes here. Okay, so you can see that from virgin to the aged material, right, the cyclic siloxane oligomers are going to decrease. Okay, and you will also see that the larger cyclic, the oligomers are going to increase here. Okay, whereas the smaller cyclic oligomers are simply going to be are going to significantly decrease as you age the material. Right, and similarly, you can see that a lot of these alcohols and oxygenated materials, oxygenated products are going to form. Right, so what it essentially tells you is, these products are formed by the breakage or scission of the polymer, right? But you can see this variation, how this happens among the different aged samples. So this says how the aging is really affecting the basic structure of the polymer. Okay, as a result of which you're getting this distribution. So it's more like a destructive characterization technique. Py-GC is a destructive characterization technique that can be used to characterize, polymers, insulation materials, and how they age with respect to different, time or radiation and so on. One more common example before I move on to the description of the different pyrolysis reactors is, those of you who are working on biomass research, right? You would know what lignin is about. Okay, lignin is a polyphenolic material, right? Its structure is not known. It will vary based on the plant source and so on, right? But typically, lignin is characterized by 3 monomeric units. Okay, one is called as a guaiacol, a syringol, and a coumaryl alcohol units. Okay, so this is characterized by 2-methoxy, 1-methoxy, or 0-methoxy groups. Okay, and typically it is known that softwood lignins contain more of monomethoxy, hardwood lignins contain high amount of dimethoxy, and grass type lignin have an equal distribution of these different linkages in the sample. So Py-GC of simple lignin can actually give you a good idea about the typical distribution of H, G, and S units in your lignin sample. Okay. So you do a Py-GC. Okay, so this is an example of alkali lignin that is purchased from Sigma Aldrich. Right. What you do is you take the material, simply you pyrolyze it at 500 degrees C. Okay, and you get a typical product spectrum. Okay, and this experiment was, by the way, performed in a Frontier single shot micropyrolyzer. Most of our works have been in that. Okay, and you get a lot of different peaks. Okay, you get a lot of low molecular weight compounds like CO, CO2, glycols, methyl glycols, and so on, right? And then what you do is you try to assemble the area percentage or even the mass percentage of all these peaks, and then try to group them as H type products, G type products, S type products, and all other products. Okay, so here you can see an example of what we have done earlier with alkali lignin, which is obtained from Kraft pulping process, and the other one is from Asian lignin. Okay, so you can see from this product distribution that the typical product distribution is the HGS distribution is very different. You can see that this alkali lignin is mostly of a softwood type, so you can see very high amount of the G type units that are present here, nearly 96. And you can see this Asian lignin, which is derived from grass, Sarkanda grass. So this contains significant amount of synapyl units. Okay, so you can use Py-GC-MS as an excellent characterization tool to even understand the typical structure or product distribution, from lignin, which is a biomass-based component. And of course, this technique has been very well validated with other characterization tools like NMR spectroscopy, which is usually used to characterize lignin due to its complex structure. So with this, I am actually moving to the second part of my talk. I'll be describing the different analytical pyrolysis GCMS systems that are available in the market. So I hope I have explained you what is analytical Py-GC-MS. Okay. Now I'll be talking about what are the different models that are available in the market. In fact, these three different things vary based on the principle by which pyrolysis occurs in the sample. So that is what I would like to focus. The capabilities of the systems, these systems are very much equivalent, but my idea is not to really promote any of these, but probably to tell you how these systems differ and how you should really look at the data that you get from these different systems. Okay. So coming to this first one, the Karthik in his first slide explained about what is the Frontier micropyrolyzer. I'm probably describing the most elementary system of Frontier, which is called as a single shot or a multi-shot pyrolyzer. Okay. So what is usually done is you take the feedstock. As I said, the particle size has to be very small so that reaction is happening in a kinetically controlled regime. Okay, you take it in a stainless steel, deactivated stainless steel cup, right? And then you're going to actually drop it inside a quartz tube that is surrounded by a furnace. Okay. Typically, the sample weight really depends on the amount of volatile matter that is present in your feedstock. So when you do any biomass or any type of feedstock, right, we do approximate analysis that gives the typical amount of volatiles in the sample, right? Typically, when the volatiles are more than 50% or 40 to 50%, you can actually take around 0.5 milligram or 500 microgram of sample. But in the case of coal, where the volatile matter is going to be very, very low, you can even take up to 1 milligram of sample. Okay. So you take the amount of sample there, you can set the pyrolysis temperature in the furnace, right? The cup gets dropped from the top to the bottommost part of the furnace. There is going to be an inert gas that is going to continuously flow through this setup. And that's going to really take the pyrolysis vapors through the capillary column of your GC mass spec. And it's going to give you a total ion chromatogram. Okay. And so this is the final data that we are interested in analyzing. Okay. So I'm going to probably focus more on how pyrolysis happens in these different systems, but not to really say what are the capabilities. Okay. So the heating rates are reported to be very, very high, and reactions predominantly occur in the kinetically controlled regime. Okay. And there are studies available in the literature which have really studied, reported the actual heating rate of the sample with different type of samples with a different type of cups, right? Exposed bead or a deep cup or a shallow cup and so on. And they have typically reported what is the actual heating rate of the sample. Because the heating rate of the sample also depends on the sample properties like thermal conductivity and other things. Okay, and so this is actually a very nice paper from the Iowa State University. So they have actually shown that the heating rates are quite significantly higher. So you can see that it can go all the way up to at moderate at pyrolysis temperatures of around 900 Kelvin, right? Your heating rates are as high as 300 to 400 Kelvin per second. And they have also carried out a lot of simulations to show that what is the typical distribution of the heating. Okay, you can see that predominantly heating occurs by conduction and convection, whereas the contribution due to radiation is quite less in this setup. Okay, again, these are studies. So this essentially tells you that how interesting is this pyrolyzer. Just because people have just studied how this pyrolyzer works, right, tells you, right, this has a lot of importance in understanding the product distribution from them. Okay, so people have actually found out what are the time constants. So you can easily say that within a second the sample is going to be pyrolyzed to the set temperature. Okay, so those of you who have used these things, you will know what are the different type of things here. So people typically deposit the sample in a hook, right, in a shallow cup, deep cup, and so on, and then try to understand what is the fundamental behind heating of the sample in these setups. Okay, the next type of reactor is a American make one. It's called a CDS Analytical Pyro reactor. Okay, so unlike the other setup in the Frontier micropyrolyzer, right, the sample is taken in a stainless steel cup, and it is more like a drop cup configuration. Whereas in the CDS, it is more like a resistive heating. Okay, so this is, you can see here a quartz tube. Okay, you take the sample inside the quartz tube. So the brown color thing that you see here is the sample that is being sandwiched between the quartz wool on either sides. And you take this sample inside this quartz tube and insert it into a platinum filament. Okay, that is resistively heated platinum filament. Okay, so based on the set temperature, right, you can actually you apply the current to the platinum filament, and that really imparts the heating to the sample that is inside a quartz tube. Okay, and of course this has high pressure operation and so on. I'm sure Frontier micropyrolyzer also has a tandem reactor that Karthik mentioned that can actually do high pressure operations and everything that this system can also perform. So this is how it is. So you have a pyrolyzer unit, and then the vapors are actually taken through a long a long 1/16 inch SS tube. Okay, a heated transfer line, whereas that is actually not there in a Frontier micropyrolyzer, and that's actually an advantage sometimes when you can prevent some secondary reactions. Okay, and you take a biomass, you can see a typical product distribution like this coming out, and then you do a typical analysis of this. One advantage of this system is, it also has a pyrolysis FTIR probe. So that means when we talk about analytical pyrolysis systems, right? The time-based information is something that is very difficult to attain. It can be obtained in a Frontier pyrolyzer, but that requires some special applications, special operations. So in this case, you have the same probe can actually be placed in the beam path of an FTIR setup, right? This is called as a Brill cell, and then as the pyrolysis happens, you can actually find out how the pyrolysis vapors are evolving with time. Okay, so here you can see an example where the FTIR, complete FTIR spectrum of the vapor phase products from cellulose and polypropylene are depicted. Okay, so each of these curves correspond to a specific time, which is typically 1 second. Okay, it increases from 1 second and then it decreases. I'll show you more data about it. Okay, it doesn't really give you the exact identity of the product, but it gives you an information about what functional groups are increasing with time and how they are increasing with time and so on. So that's an advantage of PyroProbe FTIR. So we have actually done some studies to understand what is the exact heating rate of the sample inside a PyroProbe. So these are based on some simulations that we did. And what we found out is, let's say if you take a polymethyl methacrylate polymer sample, right? The typical heating rate is not quite high as claimed by the manufacturer. Okay, you can see here that so these are the quartz tube walls. Okay, these are the quartz tube walls, and the sample is going to be placed inside, and you can see a typical distribution of the temperature along the radial distance of the sample. So you can see that if you set 500 degrees C, okay, that is 773 Kelvin, right? It would take typically 3 to 4 seconds to reach the set temperature. So that means the heating rate is not as high as 300 or 400 Kelvin per second, but typically of the order of 150 to 200 Kelvin per second. Reproducibility, I'll come to that when I show the comparison for algae pyrolysis. Okay, the third type is called as a Curie point pyrolyzer. This was actually developed even in the 1960s, but it isn't really popular in the market because it doesn't have great capabilities as the frontier micropyrolyzer or the pyroprobe reactor. So the principle here is this, okay? When a radio frequency is applied to a ferromagnetic coil, it reaches its Curie point temperature, okay? So what we typically do is the sample is not taken in a stainless steel cup, or it is not placed inside a quartz tube, okay? What is done here is we have some special materials called as pyrofoils, okay? And what is this pyrofoil? The pyrofoils are essentially some alloys, okay, which exhibit a specific Curie point temperature. That means if my pyrolysis temperature has to be 500, I have to choose a pyrofoil whose composition is Fe40 and nickel 60, right? But now if I have to go for another pyrolysis temperature, I have to choose another a pyrofoil which has a different composition, but it will exhibit a Curie point temperature of 650. So by this you can see here these are the typical pyrolysis temperature. That means these are the typical materials, okay, whose Curie point temperatures are given here. So one disadvantage is you cannot actually set the temperature of your choice. You have to set the temperature based on the Curie point temperature of the pyrofoils, okay? So typically you take the sample, okay, it can be a solid or a liquid, right? You put the sample inside and then you crimp it. That means you simply press it and then you put it inside the chamber here, and then you apply a radio frequency, right? And because this is a ferromagnetic pyrofoil, it becomes paramagnetic when you apply radio frequency, and then the Curie point temperature is reached. Curie point temperature here is nothing but the pyrolysis temperature, the desired pyrolysis temperature of the sample, okay? So it is reported by the manufacturer that the heating rate is quite high, okay, because the sample is going to be in intimate contact with the pyrofoil in which it is going to be packed, okay? So let's say if you set 764 degrees Celsius, right, corresponding to this pyrofoil, it is reported that the set temperature is reached within 0.2 seconds. Okay, so heating rates can be quite high in a Curie point pyrolyzer, but the disadvantage of this system is you can only do single temperature pyrolysis. You cannot do heating rate ramp, or you cannot do high pressure experiments or catalytic experiments and so on. That is one of the disadvantages of this setup. And of course, this is not really commercially reached the market or the users like the frontier micropyrolyzer or a pyroprobe. Okay, so here I am actually giving a comparison of the three different pyrolysis setups. Okay, so micropyrolyzer, it's a ceramic heating. I've shown that it heats by conduction and convection. Pyroprobe is resistive heating, and this is inductive heating due to RF coils, right? And the pyrolysis time can be varied, right? The temperature range is quite equivalent in all. But coming to the heating rates, okay, the manufacturer claim heating rate is more than 1000 degrees C in the case of micropyrolyzer. In the case of pyroprobe, the maximum heating rate of the filament is 20,000 degrees per second, but the actual heating rate of the sample is typically 150 to 200 degrees C per second. Whereas here it is well proven that it can reach up to 500 degrees C per second. Such data is not available for the Curie point pyrolyzer, but the manufacturer claims that the heating rate can be very, very high. Okay, so this actually motivated us to really probe. And luckily, as I said, we have all these 3 different pyrolyzers in our laboratory. So we thought we will really study the pyrolysis of a specific material. And the material that we chose here is an algae. Okay. So we wanted to choose a material like algae and then understand its pyrolysis behavior in these 3 different systems. Okay. And this is just a motivational slide to say why algae for chemicals and biofuels. It has a very good CO2 sequestration potential, and it is very good because it can actually absorb significant amount of CO2 compared to terrestrial biomasses, and the land requirement for its growth is also very low, and it can be grown in wastewater. Okay, and typical algae biorefinery can actually produce biofuels, chemicals, gases, fertilizers, protein supplements, and so on. Okay, so algae contain proteins, carbohydrates, and lipids, unlike any terrestrial biomass. So algae pyrolysis, results in oil, bio oil. Okay, and that results, the property of this bio oil is from algae is significantly different, and in fact, it is better in many sense compared to conventional terrestrial biomasses like palm, fruit bunch, sawdust, miscanthus, grass, or wood, or anything. You can see that carbon is high. You can see that viscosity is low, equivalent to a light fuel oil. Heating value is very, very high. Okay. Only disadvantage is its nitrogen content is high because algae contain proteins, and proteins are rich in amino acids, which is rich in nitrogen. Okay. So that is the major disadvantage when it comes to algae-based bio oil. But there are ways to really mitigate this so that you can actually, even get rid of this fuel nitrogen. So that the emissions when you're going to use this as a fuel is going to be lesser. Okay, so this is actually captured the attention of a lot of researchers, and we have also been working a lot on algae to fuels, both microalgae as well as macroalgae, to produce fuels and chemicals from biomass from algae pyrolysis. So it's really a hot topic. Okay. So quickly, I should be able to complete my presentation in 5 minutes, I think. So this is a typical characterization of 2 different algae that we took for this study. The idea is use all the 3 different pyrolyzers and then see what is a typical change in product distribution when we use these algae for the experiments. Okay. So we use 2 algae. One is Chlorella vulgaris and the other one is Arthrospira platensis. It's also called as spirulina, which is a very good protein supplement. It has very high amount of proteins in it. Okay, and this is a typical, we do a typical proximate analysis, elemental analysis. We also get the data on biochemical analysis and calorific value. So you can see from the thermograms, TGA thermograms, that the typical decomposition regimes of this algae are very, very similar. Although the product distribution from them is going to be very different because of the different biochemical compositions of these 2 different algae. Okay, so you can see that the basic amino acid profiles of Chlorella vulgaris and Arthrospira platensis are significantly, they are to some extent different. Okay, so Chlorella has more chlorophyll than Arthrospira platensis. Okay, so with this basic characterization, we actually went ahead and pyrolyzed these 2 different algae samples. It's a simple pyrolysis experiment at one specific temperature without the use of any catalysts or anything. And our idea is repeat the experiments and then also perform a detailed characterization of the products. Okay, so this is the data for Arthrospira platensis, spirulina algae. So you can see that the temperature that is adopted here is for all the 3 different pyrolysis setups are the same. So you can see that the results, okay, are significantly different from these different setups. Okay, so you can see that firstly, you can see that the carboxylic acids are significant from the micropyrolyzer, whereas in the Pyro probe and Curie point, we see significantly low carboxylic acids. So generally, carboxylic acid is produced, the carboxylic acids are actually present in the form of lipids in the algae. And when you pyrolyze the algae, they are simply released in the gas phase. Okay. So this is an indicator of the primary pyrolysis product from the algae. Okay, so this clearly shows that, in a frontier micropyrolyzer, at least it partially shows that in a frontier micropyrolyzer, the secondary reactions are significantly inhibited. Okay, coming to the hydrocarbons, you can see that hydrocarbons are again cracked products. So when a carboxylic acid undergoes decarboxylation, that means the release of CO2, it becomes a hydrocarbon. So you can see here that the Curie point pyrolyzer gives a significant amount of hydrocarbons. That means the carboxylic acids are getting cracked to form hydrocarbons. Okay, you can also see a lot of nitrogen containing compounds being retained, like amides, amines, nitriles, right? All these are being retained in the products from micropyrolyzer. Okay, and what you see from a pyro probe is the significant production of phenolics, okay, and cyclic nitrogen compounds. Okay, so these are typically the secondary decomposition products, okay, of any biomass or any component. Okay, so this is also evident from the char yields. Okay, so you can see that in a pyro probe we get significantly high char yields. Again, char yield is an indicator of the of the secondary decomposition. As I said in my initial slide, right, secondary decomposition can lead to the formation of more amount of solid residue, and that is evident from the pyro probe result. Okay, and if you go into the details of the hydrocarbon distribution, so you can see that so this is a typical distribution. So you can see aromatics are more from pyroprobe. Again, this corroborates the fact that phenolics are also aromatics, but you can see that aromatics generally are going to be higher from the polycyclics, the aromatic hydrocarbons. Okay, and these are aromatic oxygenates, right? So those are significantly high from a pyroprobe. Right, and you can also see here, this is another evidence that the heating value of the products are actually low from a micropyrolyzer. That means, right, the primary products are not really suffering the secondary decomposition reactions, and hence, right, most of the primary pyrolysis vapors are captured in the GCMS from the micropyrolyzer. Okay, you will see the same result reproduced here for chlorella vulgaris, of course with a different composition. So you can see carboxylic acids are very high, from chlorella vulgaris. You can see hydrocarbons are significantly high from a Curie point pyrolyzer, more than micropyrolyzer. You can see that phenolics and cyclic nitrogen containing components are high from the pyro probe. Okay, and here also you can see that aromatic components, right, aromatic hydrocarbons, polycyclics, and the aromatic non-phenols are actually very high from the pyro probe. Right, and you can see the same trend, typically, from the products. Okay, and the char yields, you can see that it is significantly high in the pyro probe. So what it essentially concludes is, right, our objective has been to compare the product distribution from the different pyrolysis reactors, and in turn understand what are the typical reactions that these algae components undergo during pyrolysis. Okay. So let me quickly go to these transformations. So you can see that typically an amino acid undergoes a lot of different reactions like decarboxylation, deamination, cyclization, aromatization to form phenols, benzene, right? Cyclic nitrogen containing compounds. Okay, amines, and amides. So amides can undergo dehydration to form nitriles. Okay, so these are typical reactions of proteins present in the algae. If you consider lipids are typically triglycerides or long chain fatty acids, right? So typically thermal deconstruction results in the formation of carboxylic acids, and carboxylic acids can undergo decarboxylation, release of CO2 to form hydrocarbons, or the carboxylic acids can undergo amination. Okay, because ammonia is going to be a major product from protein pyrolysis. Right, and this amine can actually react with carboxylic acid to form amides, and amides can undergo dehydration to form nitrites. Okay, so these are the typical reactions occurring over here. And coming to the transformation of carbohydrates, right, you can think of typically starch or a cellulose, okay, repeating units of glucose. Okay, so these can undergo glycosidic bond cleavage and dehydration to form This is actually one of the major products. It's called as levoglucosan, dehydrated anhydrous sugar, right? And carbohydrates can form indole, pyrrole, pyridine type of compounds when they react with the amine-based compounds from the proteins. And these can also undergo dehydration to form ketones, aldehydes, and a lot of these different products. Okay. I'll take up the questions once I finish the presentation. So, if you look at the typical time evolution of functional groups, right, this is also a very important information, as I said, from the Pyro probe. This is only from the Pyro probe. So you can see that generally protein-rich algae actually decompose quickly. So you can see that the evolution of the pyrolysis functional groups occur in a very short period of 5 seconds in the Pyro probe. Whereas you can see that these evolve at much longer time periods of around 10 seconds, in other type of algae, like Chlorella vulgaris. So Chlorella vulgaris, you can say, is a lipid-rich algae, whereas spirulina, the Arthrospira platensis, is a protein-rich algae. So again, based on the biochemical composition of the algae also, right, the time evolution of the products are going to be significantly different. And this can be understood from analytical pyrolysis reactors. Okay, so coming to Karthik's point about what causes secondary reactions in a pyro probe. I can quickly go. I'm almost done with my presentation, so I can take up the questions then and there. So yeah, so this is a typical construction of the pyro probe, right? So in my opinion, there is a lot of heated transfer lines and heated paths that the pyrolysis vapors have to undergo in a pyro probe reactor. Okay, I don't know if this is the exact reason, but this is what I think can be a reason. And another thing is there is always, the heating rate is not as fast as in a frontier micropyrolyzer or a Curie point pyrolyzer. As a result of the vapors have to really go through a lot of stainless steel heated transfer lines in the pyro probe, which I think is a reason for the typical secondary reactions that are occurring in it. Okay, and again, even here you see here, a lot of the C5 to C10 hydrocarbons are aromatic type hydrocarbons that you see significantly from the pyro probe. This is a typical, more detailed understanding based on the carbon number distribution of the products. So you can see that the squalene, neophytadiene, phytol, right? These are long chain hydrocarbons that are present in the lipid fraction of the algae, and these are very much retained in a Curie point as well as the frontier micropyrolyzer. Okay, so these are essentially greater than C17 products. Okay, whereas in the pyro probe, again, due to the secondary reactions, you get a lot of these aromatic hydrocarbons in it. Okay. So probably my penultimate slide. So here again, so I've been comparing the different analytical pyrolysis reactors, and again, in this just one slide, I'll show you what happens if you're really going to go for a large mass of sample. Okay, so we also have developed these microwave pyrolysis reactors in our lab, and in one study we compared the microwave pyrolysis and analytical pyrolysis of three different macroalgae. Okay, and what we found is, again, in the Curie point pyrolysis, I think this is valid both for the frontier micropyrolyzer also, right? You will see that significant amount of these anhydrous sugars are formed, right? As I said in my previous slide, anhydrous sugars are components like levoglucosan, which are the primary pyrolysis products from the carbohydrates, and these are very much retained in an analytical pyrolyzer, whereas these are nearly absent in microwave pyrolysis because, right, when we take something like 10 to 20 grams of sample in the microwave pyrolysis reactor, right, it suffers a lot of secondary reactions as the vapors spend a lot of time. So you can see that anhydrous sugars have decreased and furan derivatives have significantly increased because anhydrous sugars can decompose to form furfural, HMF, and methylfuran type of products. So this is also, it's more like a very general observation found across different type type of samples. To one of the questions from Adriana, have you researched other biomass in both Frontier and Pyroprobe? We are doing it. We have not. So this is our first study with algae, and we will be doing more type of biomasses. And these are essentially, the seaweed type of samples, Khurshid. So the Turbinaria, Sargassum, these are essentially seaweeds collected from the Mandapam camp near Tamil Nadu. Okay, so that's my summary. So what I have described in the past 50 minutes or so is, analytical pyrolysis GCMS is a very powerful tool to characterize a wide variety of materials, and also nowadays it's more widely used based on the papers that are being published in journals. It's more widely used for biomass pyrolysis research and small sample mass. There is some fundamental that really goes into that, right? Small sample mass ensures that reaction occurs in a kinetically controlled design. And mode of feeding plays a decisive role. That is what we observed in our study with algae pyrolysis. So we found that generally the Frontier or the Curie point pyrolyzer results in much faster heating rates, and the secondary transformations are quite low in it compared to the Pyroprobe. And these are some of our results, right? Carboxylic acids were significant. That's an indicator of the primary pyrolysis products, right? And secondary reactions do occur in Pyroprobe. And we have independently done a lot of studies on biomass pyrolysis in Pyroprobe, and we did find that the secondary products, typically aromatic hydrocarbons, are significantly high from it. Okay. So that's my acknowledgement to my students. Many of them are also attending here. I think they have done most of these studies. Big thanks to them. And thank you for your attention. I can take up the questions that are in the chat screen. I think I did answer some of the questions.
Yeah, thank you, Prof. For the wonderful lecture. I think some of them have been they're typing their questions. Others, please take your time to type your questions while Vinu is answering these questions. Yeah, I think there are a couple of questions that you have not seen it yet. I think that they are on the top. Maybe if you can read, or I can read for you.
No, I can go through. I have another screen. So
okay. Yeah, the book is available free. It's not free. So yeah, I think you can buy through Amazon. I think this book is not available.
I have a copy. I even bought it. I think it's a very nice book, actually. I mean, it has like a wide variety of synthetic polymers. So before really trying a commercial polymer, it will give you a very good understanding of what are the products you can expect from a typical or a virgin polymer sample. And based on that, you can actually make your further hypothesis. So how to form a kinetic mechanism by using these product distribution? I think this is, based on, you can do these experiments at multiple temperatures, see how the product distributions vary with respect to temperature, and then try to propose some mechanisms. But mostly it depends on using multiple analytical techniques to arrive at the mechanism, because these kind of algae or biomass are very, very complicated components, and the products are significant, actually. So you will get in the bio oil fraction or biofluid fraction, you will have 50 to 100 different components in it. So you can probably try to understand the mechanism of formation of the major compounds. That is what is very much possible. So Curie point is supposed to be fastest heating. What causes secondary reactions? So that was Karthik's question. See, generally what we found is, the major difference that we found between the Frontier micropyrolyzer and the Curie point pyrolyzer is typically in the the hydrocarbons and carboxylic acids. So what we found is we see that consistently the carboxylic acids are easily getting decarboxylated to form hydrocarbons in the Curie point pyrolyzer, whereas that is not significantly happening in a micropyrolyzer. Okay. it may be because I don't know. It's a hypothesis. It may be because because the sample is going to be in very much of an intimate contact in the case of with the pyrofoil in a Curie point pyrolyzer. Maybe it suffers a very high temperature. For significantly long duration. By significant, I mean probably for a few more seconds that is leading to the formation of these hydrocarbons. But otherwise, except this, we do find that majority of the product distributions are sort of similar between the micropyrolyzer and the Curie point pyrolyzer. So which heat transfer mechanism is influential? So conduction or convection? So again, this is a very important question, and it actually corroborates, it can actually corroborate the previous question also. So in a Curie point pyrolyzer, I would expect that conduction is the predominant mode of heat transfer, whereas in a micropyrolyzer, as it is also shown by the Iowa State researchers, it is both conduction as well as convection. Okay, I can quickly go to my this slide. So they have shown that it is typically 50-50 convection and conduction in the case of a furnace type micropyrolyzer, whereas in a Curie point pyrolyzer, I would say that there is no possibility of convection because the sample is simply packed within the pyrofoil plates. So it is predominantly conduction, and that may be a reason. Conduction may be more dominant in a Curie point pyrolyzer. That's actually a good point. Mr. Baburov, waste engine oils. Yeah, I think we have discussed earlier. I think so. One of the points with waste engine oils is that vaporization of the sample is going to be significant when you apply such high temperatures to waste engine oils. So you cannot expect that the entire material is going to pyrolyze or crack, but once you expose the oils into pyrolysis system, you will actually see a lot of these components, long chain hydrocarbons, simply vaporizing due to temperature. Right. So vaporization is different from devolatilization, because devolatilization is simply due to cracking of the material, whereas vaporization is simply a boiling effect. So that will be significant in the pyrolysis of waste engine oils. Different masses. So because all these systems are connected with GCMS, all these are pyrolysis GCMS systems, there is always a limitation on the mass of the sample that we can use. Typically, we end up using lesser than 0.5 milligram or 500 microgram of sample. So we cannot go beyond that, because in that case, you cannot actually send a lot of vapors through a GCMS system. Either you have to increase the split ratio, or the conditions can typically vary. So in these kind of systems, going for large masses is not really recommended. But certainly when you use it, certainly you will see different results because of the mass transfer and heat transfer coming into the picture to a certain extent. And these are significantly different from a phaser reactor. So phaser reactor developed at University of Minnesota by Paul Downer. It's actually advanced in the sense that you can actually track the time evolution of specific masses in a phaser reactor. And so the whole concept of this is also quite different. Concept of how you really pyrolyze the sample is also very different. So it is certainly different. And in fact, those reactors are phaser reactors, very much useful to understand, to really do the kinetic simulations, right? Most of the data from phaser reactors can be very much used in the kinetic simulations to understand the the reaction, the exact reaction mechanism of specific reactions with model compounds that can be done very well in a phaser reactor. Is there any, if I have missed any question, maybe Karthik can tell me, but
yeah, I think Shubhan, you want to ask some question, so you can turn on your microphone. Ask.
Hello, sir.
Yeah, tell me.
Hello, sir. First of all, thank you for the nice presentation. Sir, my question is, sir, for the analytical techniques by GCMS. So, after pyrolyzing a sample, instead of sending the volatile matters to GC, if we will send it directly to MS, is there will be any problem?
No, it can be done, no doubt about it. In fact, I mean, people have really done this, and the advantage that you get by sending the vapors through a GC is the components are going to be separated through the GC capillary column, and you get the identity of individual components, right? The MS will really do the comparison for you, and it will tell you that, this is the likely component. But if you are going to directly infuse the pyrolysis vapors to a MS, you get all the masses that are going to be present in the vapors without any separation. So you have to use your understanding of the basic chemistry of the reactions to find out what each of these peaks would correspond to. So that is the basic difference. But it can be done, no doubt. If your feedstock is not as complicated as a biomass or an algae, but if you're using a simple single molecule component, that can be done. And if you know what is the typical product you would get out of it, it can be done.
Hello.
Okay, sir. Thank you.
Yes.
Thank you very much, sir.
Yes,
I can hear you.
I have just one question.
Yeah.
When you do actual pyrolysis, because I have used frontal macro pyrolyzer, so when you do the pyrolysis and your volatiles go to GCMS or GC or GCMS, you unidentified product in the product stream. So how to tackle those? Once you try to match with the GCMS data, it will give very hazardous compound. You really cannot correlate with the initial composition, and it is very difficult to choose those compounds. And at the end of the you will you will end up there by saying there's unidentified product. So how to encounter those products also as a product stream?
Okay, that's a very nice question. So it is difficult. I mean, first I have to acknowledge that it is not possible to get 100% of the identify 100% of all the products. Always in any product distribution that you find in the literature, you will see some others or other components which are identified with a very low match factor in a GCMS, or whose composition is going to be very, very complex. So you have to resort to other techniques. So you have to one way is that cannot be that people have done some things like this. You have to instead of connecting the pyrolysis vapors through the GC column, you can simply condense the pyrolysis vapors in a chilled methanol bath or something, right? And then you take the compounds and then analyze it in a GC in GCMS as an offline thing, and then try to identify all these different components. So sometimes people use alternate methods like LCMS to identify the exact components that are present, unidentified fraction that we usually report in the GCMS. So that we have to go by using other techniques, and GC cannot completely identify all the components that are present. Typically, things like dimers, trimers, those will be very, very difficult to characterize using the GC. So people go for LCMS based characterization, and this is a major problem, especially when you talk about lignin, because in lignin, a majority of them, large molecular weight components, in fact, stay in the char or the solid fraction. So again, you might have to take the char, kind of dissolve it in a solvent, and then inject it. So you have to resort to multiple techniques. So that is my answer. But we try to close the mass balance to the extent possible by using some pure components, or we can calibrate them by using pure components, and try to use the calibration equation to calculate the k factor for other components and so on. We can do all these things, but still there is always an unidentified fraction which we need to resort to other techniques. Either we condense them in a liquid fraction, and then we can subject it to LCMS or NMR or other techniques, and we have to do that.
One last question, I think, is from Pawan. So with that, I think we can end it. So
fossil fuels are not formed by pyrolysis. Pyrolysis. When we say pyrolysis, typically we refer to heating at high temperatures, and typically we are not really talking about the pressure, which is typically, but it is assumed that it is mostly atmospheric pressure. So fossil fuels are typically formed due to a high pressure process similar to hydrothermal liquefaction conditions, and not due to pyrolysis conditions. So oxygen molecules come in biocrude because there is significant amount of oxygen present in the natural matter itself, that we get out of it. I think Pawan, we can discuss about it later through email if you're not really clear about the answer.
Yeah. Okay. I think, if you don't have any further questions, so please join me thanking Prof. Vinu for his wonderful lecture, as well as the answers for different questions we had. So thank you, Prof. Vinu. And
thanks, Karthik. Again, a big thanks to Karthik and Frontier Lab. I think this is an excellent initiative. We really plan to continue this. I think in the next months we will identify a speaker, and we will let you know the link, so that you can attend the pyrolysis lectures on multiple topics.
Yes, that's correct. So I also would like to thank all the participants for very enthusiastic participation, asking questions, and being interactive. So thank you all, and let's close the session and have a good evening. Bye.
Thank you. Thanks all.
Vinu-san, how are you?
Fine. I'm fine.
Yeah, I'm fine. It was a very informative presentation. Thank you.
Thanks, Anusha. How are you doing?
Yeah, presently safe and fine for now.
Okay.
Don't know what's
You're in
going to
India,
happen
right?
in future. Yeah, in India.
Okay.
In Vizag now.
Oh, okay.
So how is the situation in campus? Fine.
It's we are slowly getting back to work. Students are slowly coming back. Not everyone has come back. Those who have got the e-passes to travel to Chennai have come back, and some of them are in quarantine. Yeah, it's going on like that.
Okay. Okay then. Thank you, Vinu-san.
Yeah, see you. Bye.
I'll take a leave then.
Yeah,
See you again.
I'm also signing off.
Good afternoon, Professor Vinu. I am
Good
Babu Rao.
afternoon. Hello, sir. How are you?
I have a question which is not a biomass pyrolysis, it's a pyrolysis. I came purposely to meet you on the end of March. Unfortunately, the lockdown starts and we are unable to move. And then I also communicated to your office number, unable to reach you.
We
So,
have your I have your samples. I have one of your sample, but again,
yeah. Thank you for that. I will be flying back once the lockdown is completed and the flights are flying back. I'm planning to come back on December again. Possible
okay,
for me
sure.
to have your time to so we can work on a project to bring the black material to a proper diesel material.
Sure, we'll do it. We'll do it. Right. In the
Do
meanwhile,
you have a catalytic method? You promised to Dr. Venugopal that you have a